All right, welcome back to semester 2 of Anatomy and Physiology Lab. So usually we do a whole bunch of dissections in the spring, but we're going to stick to this complete anatomy app and see how it goes here. So same drill as last semester. You can go onto the complete anatomy Canvas page, and up there you can see that you have your structures list. And in fact, I'll just share my screen with you real quickly. So if you go up on into Canvas, this is what you're going to find. You're going to find your structures list, not too many for cardiac. And we'll work through, most of the time we don't work through most of these structures. I think we'll touch on most of these today. So that'll be nice. And then the introduction sheet that Manny's put up here as well. So for example, it says here, identify your structures as usual. And then with incomplete anatomy, we can find some stuff by using this prefix HRT. So let me just demonstrate that. All right, so if we go in here, there's a couple different ways to do this, right? You can go to your courses and then you can click on Spring 2021 for the lecture that Manny has prepared. Or we can go back to, let's say, Atlas. And then if we search for that prefix HRT, just for heart, it'll bring up all of the individual slides that Manny has prepared, and then you can review these. And we'll have hyperlinks to these today as we work through the PowerPoint as well. Okay. Some other things to point out on this intro sheet. We'll go through the PowerPoint. Oh yeah, your review quizzes and everything like normal. So if we go here.
Oh, girl, no, you're totally good.
You could mute, that'd be awesome. So here's the lecture that you can work through. Here's your heart anatomy review quiz. And then remember, do not touch this heart anatomy practical until you are ready to complete it and have that serve as your score for that practical. I think you're all familiar with that now. Some other things Manny pointed out here on this intro sheet. There are videos that he selected that are helpful. So if we go back to, let's say here, we go up to videos. He's pulled some here that he thought would help you. So for example, we can go down to, here's borders. I think this one's about a minute long.
If we were to take a look at the heart from a purely anterior or posterior perspective and draw boundary lines surrounding it along all four sides, what we would see is that the heart is approximately trapezoidal in shape, with borders on both the right and left sides and a superior and inferior border on the top and the bottom. Each of these borders is going to consist of different structures within the heart. So let's now take a closer look at each of these borders independently, beginning with the right border of the heart.
These are all
the right border of the
list.
heart is formed by the superior vena cava, the right atrium, and the inferior vena cava. The left border of the heart is going to be formed by the left auricle and the left ventricle of our heart. The superior border of the heart is going to be formed by the superior vena cava, the ascending aorta, and the pulmonary trunk. And lastly, the inferior border of the heart is going to be formed by the right and the left ventricles. Now we're going to take our time to look at all of these structures in more detail.
Yeah, and we will look at them again here shortly, but I thought that was a good introduction, and those are some structures now you can just cross off your structures list that you know, and they're named kind of accordingly, not too bad. Manny also wanted to point out there are some radiology slides you can take a look at. Let's see. And these will help, they helped me probably most with your vessels, which can be a little tricky, the vessels of the heart. So he's pulled those slides again or select ones. You can work through those So we'll go to radiology. What I think, yeah, coronary angiogram. This is a good one. Okay. Then for example here, we can look at, and it'll highlight it or in the structure here that you can rotate on the heart itself, and then it'll show you the actual image here. So that's your right coronary artery, for example. So those students have found those helpful as well when we're working through this. Okay. Just wanted to point those things out. And then I think without further ado, then we can begin PowerPoint, and then we'll take questions at the end if that sounds good. All right, so we'll roll through this PowerPoint and we'll touch on a lot of structures that are on your list of structures you need to know. And then pay special attention, we'll have that complete anatomy hyperlink down in the corner of some of the slides, and that'll allow you to access whatever we're talking about in that specific slide directly in complete anatomy. All right. The heart's found within the thoracic cavity, and it's surrounded by the thoracic cage, or what we call the rib cage. And within that thoracic cavity and within those ribs, it's surrounded by the lungs. And it sits anterior to the spine. So it's in front of the spine, but behind our sternum. So we have this bony protection on all sides, which is great for protecting this vital organ. We'll see that it sits a little bit left of the body midline. So if I just go ahead and sketch the body midline there, we'll see that it lies a little bit left of that. And we also see that it's slightly rotated. So this left side is going to be more anterior forward, and then this back side, or the left side, is going to be rotated posteriorly a tiny bit. If we look at some surface features of the heart, we have the base of the heart, and this is where our vessels attach to the superior part of the heart. This is also called the superior border, if you think of the complete anatomy video that we watched before starting this lecture here. So we got the base of the heart, and then we have the apex of the heart, and that's that conical end. So some people tend to think this looks like a ice cream cone. You have that conical tip that points a little bit inferior. And in fact, that conical tip, when we get a contraction of the heart, it hits the inside of the chest wall, and that's why we think our heart primarily lies on the left side, and we put our hand over our heart on the left side of our chest because it's hitting the chest wall there with each contraction. We also have our other borders that we mentioned as well. So we have that inferior border. Remember, it makes that trapezoid shape. And then we have that right border and the left border. I have thrown a few extra slides in here just for those of you who don't have A&P lecture within Block A, kind of make a little more sense of this anatomy. We say that the heart is a dual pump. So we have the right side of the heart and the left side. And they act as separate pumps. The right side is going to be dealing with deoxygenated blood, and the left side is then going to be pumping oxygenated blood. And we see that it's divided into 4 different chambers. Okay, so the atria are the upper chambers, and the atria are the receiving areas. Think of an atrium where everyone gets together and greets and is received. Right? So this is where blood is going to return to the heart, whether it's from systemic circulation or from the lungs. And then the ventricles, these are the powerful areas of the heart that pump the blood out to other areas, whether it's being pumped up to the lungs or out to systemic circulation. And then we have septum of the heart. So this divides the different chambers of the heart. So we have an atrial septum, divides those atria, and then the ventricular septum that divides the ventricles. We don't want to be mixing deoxygenated with oxygenated blood, so these are important. And we have great vessels associated with the heart. These are great because they're large, and they're what transport blood to and from the heart's chambers. So we start with incoming deoxygenated blood from systemic circulation. That blood from the upper part of the body is going to enter the superior vena cava. Vena cava, Latin for hollow vein, large hollow vein, which is in fact what it is. And then we have an inferior vena cava as well, and that'll enter into that right atrium from the lower portion of our body. Okay. So that'll be then pumped to the lungs. Now, we tend to think of oxygenated blood as always being found within arteries and deoxygenated always within veins. And that's not always the case, as we'll see here. And in fact, it's better to think of it as arteries always transporting blood away from the heart, while veins transporting blood toward the heart. Because if we move on now, and let's say we pump this blood from the right atrium to the right ventricle, well, then it's going to be pumped up into these pulmonary arteries. So here's the pulmonary trunk, for example, the pulmonary arteries. And when it's leaving the pulmonary arteries, although the blood is moving away from the heart, so it is an artery, it's not oxygenated yet. It's on its way to the lungs to get oxygen, but it still does not have oxygen. So this is one of those exceptions to the rule where we have deoxygenated blood found within an artery. Okay, then we'll return to the heart via these pulmonary veins, and we'll enter into the left atrium, and then we'll be pumped into the left ventricle, and then out the aorta. And the heart is actually enclosed within 3 layers. So there's one outer layer, and then there's 2 inner serous layers. And serous because they secrete serous fluid. It's kind of like a lubricant, if you've ever heard that term before. So we'll start with the outer layer, the fibrous pericardium. I should mention here that we call it the pericardium, peri meaning around, cardia, heart. So around the heart, which is where these layers are found. So the fibrous pericardium encloses the heart. That's that whitish fibrous layer shown here. And if we zoom in on a section of this heart and look up here, we can see that outer fibrous pericardium a little bit better. Okay. So this encloses the heart, though it's not directly attached to the heart. It's kind of like this bag that surrounds it. And that prevents chamber overfill. So it kind of acts as a barrier to, those chambers distending with blood entering them. It's going to keep them in check. And then directly adhering to the fibrous pericardium, we have the parietal layer of the serous pericardium. Parietal just means wall. So it's like the wall layer of the serous pericardium. It's a little bit tricky to see, again, but if we zoom in on our image up here, we can see our outermost fibrous layer, this white fibrous material, and then directly beneath that, I'll switch to a different color, adhering directly to it, we can see that parietal layer of the serous pericardium. So they're stuck together. And these first 2 layers we've discussed, the fibrous pericardium and the parietal layer of the serous pericardium, they form what we call the pericardial sac. Okay. Now, usually we do a frog heart dissection when we meet in person during a regular year, not block schedule. And the first thing we do when dissecting this frog heart is we have to snip and remove the pericardial sac so that we can actually get to the heart inside of that sac. This is a protective sac, helps with lubrication, protects the heart from rubbing against other structures. Then our final layer of the pericardium is this visceral layer of the serous pericardium. Visceral like viscera, so this means directly adhering to this organ. And in fact, we can kind of see here, again, if we zoom in up here, while we have our first 2 layers, the fibrous, then the parietal directly beneath that adhering to it, then beneath the pericardial cavity, we have the visceral layer of the serous pericardium. Okay. Also sometimes called the epicardium, epi meaning upon. So this is directly adhering to the heart now, where the other 2 are not. They form like an external bag, that pericardial sac. This one adheres to the heart. And so we see then that our 2 serosal layers, our parietal layer and our visceral layer, they form this pericardial cavity. There's this open area between these 2 layers, between those 2 layers. And remember that these are serous layers, so they secrete serous fluid. And it enters that pericardial cavity. That's where we find that serous fluid, these secretions from the epithelial cells of those serous layers. So as I'm apt to do, I have a balloon analogy here for you. So if you were to take your fist and put it into a balloon, this is going to kind of represent those different serous layers of the pericardium. So you'll have your innermost layer adhering directly to the heart. the visceral serous pericardium, and then you'll have the outer layer of your serous pericardium, the parietal serous pericardium. So they form this like double-walled area where we can fill the inner space with serous fluid. And that serous fluid acts as a lubricant to help prevent friction with each contraction of the heart. And then outside of all that, we would have our fibrous pericardium that would be adhering directly to this parietal layer. And I guess before we move on, I should point out that we do have a complete anatomy hyperlink for you here within the PowerPoint. All right, I'm going to show you some more superficial anatomy. Let's begin with the coronary sulcus. So remember that a sulcus is a deep groove. And coronary, actually, I guess relating to a corona, which is a crown, and that's because this coronary sulcus kind of follows the circumference of the heart. So it's going to start here and then wrap all the way around the back and around here. So it's like a crown or corona that this heart is wearing, hence coronary sulcus. And we'll see that the biggest vessels of the heart travel here. And they're usually covered within fat. We can see these fatty deposits, so they're a little hard to see, but we've drawn them here with the fat removed. So that's our coronary sulcus. We can see that our coronary arteries are found within that sulcus. And then we have the interventricular sulcus. So it's easier to remember because it's interventricular, which means between the ventricles. So we have this deep groove found between the ventricles. And again, we have vessels found here. One other thing I'll point out that we can see here is the auricle. Okay. So the auricle is this flap-like extension of the atrium. And we can only see it when the heart is not full or when it's at rest and it's not distended with blood. And so it just helps create more volume where blood can fill that atrium. Real quickly, we have a posterior view of the heart, and we can see then our coronary sulcus wrapping around the circumference of the heart. And now we can see that on the backside of the heart, we also have an interventricular sulcus as well, another groove between those ventricles. All right, if we turn to the internal anatomy of the heart now, we have a cross-section shown here. The first thing we'll notice is that the ventricles are much thicker than the atria. And we can see that there's a lot more cardiac muscle. Now, we need to have, the ventricles need to be able to generate more forceful contractions because we're pumping blood out of the heart, either up to the lungs or systemic circulation. Now, if we're pumping blood to the lungs, we're going to need to generate a little bit of force. So we'll see that our right ventricle, there's some cardiac muscle, it's not very thick, but look at our left ventricle. This is pumping out to systemic circulation, so we need to be able to generate force to pump this out the entire body. So it's 3 times thicker than the right ventricle, the left ventricle is. Now, the heart walls have 3 layers. They are a epicardium, a myocardium, or our cardiac muscle, and the endocardium. Let's take a look at these independently. So if we take a cross section of the wall, we can see here this would be our most external layer, and then this would be the internal layer of the heart. This is where we'd have blood interacting with this surface. So the most external layer of the heart wall is the epicardium. And we've talked about the epicardium before because it is also the visceral layer of the serous pericardium. That layer of the epicardium that is adhering directly to the surface of the heart. So this also makes up the outermost layer of the heart wall. And we said that it is made up of simple squamous epithelium and it's able to secrete serous fluids. The middle layer of the heart wall is the myocardium. Myo, that prefix meaning muscle, and then cardia, heart. So this is cardiac muscle. This is what generates the force to pump the blood And this can thicken with exercise or atrophy with lack of exercise. And then our innermost layer then is the endocardium. Endo meaning within. So this covers the internal surface of the heart, and this is actually continuous with the lining of the blood vessels. So this is going to join and then become that lining of the blood vessels as they leave the heart. There are some other structures I want to point out while we're taking a look at the internal view of the heart. Let's start with the pectinate muscles. Pectinate muscle is found within your right and left atria. And we can see here these muscular ridges found within them. If you get your hands on one of the physical heart models that we have in lab, you'll see that some of those models just omitted these, while others have nice pronounced pectinate muscles. So these muscular ridges, they just help generate more forceful contractions within the atria. All right, now for probably my favorite structure you're going to need to know, the fossa ovalis. So fossa meaning a shallow depression, and ovalis because it's oval shaped. And the fossa ovalis is the former foramen ovale. And the foramen, remember, is a hole. So within fetal development, you're going to have this hole where blood can move right from your right atrium into your left atrium through that foramen ovale. It shunts blood from the right atrium to the left atrium within a fetus, so we can bypass the lungs. So think about it. If you are a fetus, you're getting your oxygen supply from your mother. So there's no reason for us to pump blood from the right atrium into the left ventricle and then up to the lungs. Okay. So what that foramen ovale allows us to do is take that blood from the right atrium, dump it into the left atrium because it's oxygenated already coming in from your mother, and then that'll be pumped out systemically to your body. And another structure I'll point out while we're here is this trabeculae carneae. So that translates to beam flesh. Okay? And these are the muscular ridges here, or beams, that we see within the ventricles. And again, they're going to aid in allowing for a more powerful contraction of those ventricles. Next, we're going to talk about the valves of the heart. So there are two different sets. We're going to have atrioventricular valves, which connect the atria to the ventricles, and then we're going to have semilunar valves that move out to great vessels. Let's begin with the AV valves. So again, the valves help prevent backflow of blood within the heart, and the AV valves specifically are going to help prevent blood move from the ventricles back into the atria. So we have a right atrioventricular valve and a left atrioventricular valve. Okay, atria, and then ventricular, ventricle. That's what's connecting the two. So we can see the blood is going to move from the right atrium into the left ventricle, and from the left atrium into the left ventricle via those respective valves. Let's take a little bit closer look at the anatomy of these. So let's take a look at how these valves work. So I'm showing you here the right atrium and the right ventricle. Of course, these are connected then by the right AV valve, the right atrioventricular valve. And let's say we have a contraction of the atrium. Well, that is going to force blood through this AV valve into the right ventricle with ease. These flaps, the AV valve, are going to open up and blood's going to move on through. Now, when we get a contraction of the ventricle, it's going to contract and it's going to push some blood backwards towards the right atrium. And when that happens, the right AV valve, those flaps are going to snap shut. You can see here we have a papillary muscle, and the papillary muscle is connected to these chordae tendineae, or tendinous cords. And they support these flaps of the AV valve. So you can imagine that they're going to prevent these flaps from inverting with the valve, meaning that they would flip back, kind of like in this orientation, and allow blood to move back into the atrium. So because we have the support from the papillary muscles and these tendinous cords, we don't get inversion of this valve, and blood, when it's pushed back by the ventricle with a contraction towards the AV valve, it snaps it shut and blood only moves forward toward the lungs. If we were to take a kind of bisect this heart right through here, and then look at it from the top down, what they're calling a coronal section, moving through that corona area of the heart, we would see that the valves look different. So we'll see the right AV valve, it has 3 different cusps. So for that reason, it's called the tricuspid valve. We compare that to the left AV valve. See, there are 2 flaps or cusps, so it's sometimes called the bicuspid valve. The other common name you'll hear for this is the mitral valve or mitral valve, and that's because a bishop's hat kind of resembles this shape here seen within that left AV valve. So we said AV valves prevent backflow of blood within our chambers, so blood doesn't move from ventricle to atria. But we also have these semilunar valves, and they prevent backflow of blood from our great vessels. So we'll see that there are 2. There is an aortic semilunar valve and a pulmonary semilunar valve. And they're called semilunar because they thought these little cusps here look like little crescent moons or half moons. Okay. So we see that there is an pulmonary semilunar valve, and that's going to prevent backflow from the pulmonary trunk to the right ventricle. And then we have the aortic semilunar valve, which is going to prevent backflow from blood from the aorta to the left ventricle. And we'll finish up today by looking at coronary circulation. We'll start with the arteries, and I guess we'll start with coronary artery since we've talked about the coronary sulcus, where the coronary artery lies. So there's a right coronary artery and a left coronary artery. And this is what supplies the heart with its own oxygenated blood. And we'll see here that our coronary artery has branches. So branches of the right coronary artery include the post-interventricular artery. So this is a posterior view, so we can see if we wrap behind the heart here, it's running between the ventricles, so they're showing it a lighter color. And for that reason, you really need to delve into complete anatomy where you can rotate these structures. It's just going to be a lot easier to learn these vessels. Okay, so that's our first branch, is that posterior interventricular, because it's found posterior on the heart and it's running between the ventricles. And then we have the right marginal artery that branches off of the right coronary artery. So here's our right marginal artery. Okay. Then we have our left coronary artery, and it branches to the circumflex artery. It seems to kind of wrap posteriorly a little bit, so I like to think of it as flexing backwards, circumflex. And then we have the anterior interventricular artery. So this runs between the ventricles on the anterior side of the heart, not to be confused with the posterior interventricular, which runs on the posterior side between the ventricles. All right, I'll point out some of the veins here. Again, it's going to be important to get into complete anatomy so that you can both rotate these structures and then look at additional structures on your list that I haven't touched on. I'm just touching on some main ones here. So we'll start with the great cardiac vein. This will run along that anterior interventricular artery. You can see if you were to superimpose, they're going to be found within that sulcus. So this is how our deoxygenated blood is going to leave that cardiac tissue and get back to the heart where it can be pumped up to the lungs to be oxygenated. So example, if we're leaving the great cardiac vein here, we'll wrap around and then we'll enter into this coronary sinus. So this serves as a collection area for all these veins that have deoxygenated blood. So it can enter the right atrium here and then be pumped into the right ventricle and then up to the lungs to get more oxygen. So all the deoxygenated blood is going to flow into this coronary sinus, but before moving back into the right atrium. We also have the middle cardiac vein. You can see that's located posteriorly here. It's shown lighter. And then on the anterior side, the small cardiac vein. There are some more anterior veins here you'll need to know. All right, now we're going to put these structures together and relate them to deoxygenated and oxygenated blood. And we'll just kind of number them off as we move through the different structures we've talked about. So we'll begin with the inferior and superior vena cava. So superior, this is going to be deoxygenated blood returning from your head and your upper limbs. And then inferior vena cava is going to be returning blood from your trunk and your legs. And that deoxygenated blood is going to move into your right atrium. So notice the structures here shown in dark blue. That's representing oxygen poor blood. And then our red will be oxygen rich blood. So here we're talking about deoxygenated blood. So that right atrium, that deoxygenated blood, is going to move via that right AV valve into the right ventricle. Then it's going to be pumped, passing through that pulmonary semilunar valve up to the pulmonary arteries. So this is one of those goofy instances where we're talking about arteries, but it's deoxygenated blood. But if you just remember that arteries carry blood away from the heart, you'll be okay. So we go up to the lungs, we get oxygenated, and that blood then returns via veins toward the heart. So pulmonary veins lead to the left atrium. From the left atrium, we're going to move to that left AV valve into the left ventricle, and then we're going to pass through the aortic semilunar valve to the aorta, and that blood's going to move to systemic circulation. And that will be it. You're free to go. Unless you got some questions, I'll upload this and email it to you all shortly.
Oh, I have a quick question on
Sure.
the complete anatomy. Are
Yeah.
we doing it individually still? Like with the heart, or are we combining multiple? Because I know we switched it last semester, but I just want to know.
How do you mean individually?
Like just the heart anatomy, and then I don't remember all the other ones.
Yeah, so what are there? I think there's 5 or 6 different mini practicals. We kind of broke them up into smaller practicals for you. So I believe heart is up there now. I think vessels are up there as well. And we'll be going through vessels either next week or the following. It's whatever on that schedule that I sent you. I don't know if I answered your question at all, but
Yeah, okay. I just wanted to know if it was like a mini quizzes like last time, or it was like major ones. Like if we had, like 50
no
questions.
major 175.1s like last time.
Okay, sweet.
Those were just apt to crash so much and then they created problems. So,
Okay, great. Thank you.
so each one's worth 5% essentially of your lab grade.
Got it. Thanks.
Yeah.
Sweet. Thank you so much.
Yeah. Take care.
Thank you.
You're welcome.
Thank you.
See ya.
Thank you.
Bye bye, guys. We'll see you later.